everyone, welcome back to ConceptWala. In today's video session we are going to talk about endogenous pathway. So in previous video session you saw in exogenous pathway how MHC II molecule present antigen to CD4 plus T cell. Now in today's video session we are going to discuss how MHC I molecule present antigen to CD8 plus T cell. So let's just learn the whole procedure through a word diagram. It will be much easier to understand. So let me just draw a straight line first. It is not a straight line but it's okay. So this side is your RAF endoplasmic reticulum lumen and this side is your cytosol. Okay. Now every cell has a proteolytic system present in the cytosol which is known as proteasome. So when a viral protein or an antigen enters the cytosol or reaches the cytosol, the proteasome degrades the antigen. Okay. So let's just assume that this is the proteasome and let me just change the color, and this is the viral protein. Okay. Now the viral protein or any misfolded protein that is going to be degraded is tagged with a ubiquitin molecule. Now this ubiquitin molecule actually bears the signal for degradation. So this viral protein is tagged with ubiquitin molecule and now the proteasome will degrade it in oligopeptides of 8 to 10 amino acid. So these are the fragments of the antigen. Now this antigen particle or the fragments of the antigen will reach the RAF endoplasmic reticulum through tap transporter. So this is your tap transporter, and through this tap transporter the antigenic peptide will reach the RAF endoplasmic reticulum lumen. Okay. Now you guys know that the MHC I alpha chain is synthesized by the MHC gene itself, but the beta 2 microglobulin is not synthesized by the MHC gene. So the assembly and packing of this MHC I molecule requires the help of chaperons and some protein molecules. So the first chaperon that comes in the picture is calnexin. Okay. So let's just draw it here. So let me draw the MHC I alpha chain in black. So this is your MHC I alpha chain and this is your calnexin. Okay. And this is your enzymatically active protein which is ERP57. Now this ERP57 occupies the peptide binding cleft of the alpha chain. It prevents any premature peptide binding to that alpha chain, and the binding of this calnexin and ERP57 actually prevents premature degradation of the alpha chain. So when the beta 2 microglobulin molecule binds with the alpha chain, the calnexin molecule will be removed. Okay. So let's just draw in the blue. So in the green color this is your beta 2 microglobulin, it will associate with the MHC I alpha chain. Two other chaperones which is tapasin and calreticulin will also be associated with the MHC molecule, MHC I molecule actually. So this is, oh, let me just change the color. So tapasin and calreticulin. Okay. It will also associate with the MHC molecule and calnexin will be removed. So this is your MHC I alpha chain. the beta 2 microglobulin molecule, ERP57 is still attached with the peptide binding cleft and to other chaperon, which is calreticulin and tapacin. Now this tapacin brings the tap transporter in close proximity of this MHC1 molecule. So it helps to take up an antigen molecule. Okay. Now there is another aminopeptidase which is known as ERAP, endoplasmic reticulum aminopeptidase. Now this ERAP provides proper shape to the aminopeptidase or the antigenic fragments. Okay, so it will provide the proper shape, the shape that is required for binding with the MHC1 molecule. Now the MHC1 molecule will take up an antigen and all the protein molecules and associated chaperone molecules will be dissociated from the MHC1 molecule. Okay, so this is your MHC1 alpha chain and beta 2 microglobulin that makes the MHC1 molecule, and the MHC1 molecule takes up an antigen and all the chaperones and protein molecule, the ERP57, tapacin, calreticulin will be dissociated from the MHC1. Now this MHC1 will leave the rough endoplasmic reticulum lumen, so it will leave rough endoplasmic reticulum and it will reach the cytosol. Now eventually it will reach the cell surface and the CD8 plus T cell will recognize the antigen and it will destroy the whole cell that has the antigen, okay, that presents the antigen. So this was the last part of our MHC series. You guys can download the lecture PDF from our website, that is www.conceptwala.com, or I will put the link in the description box. Go check out our website for course details. You guys can join our courses or just buy the lecture PDF from our website. So be with us, keep watching Concept Wala and keep learning, keep growing. Thank you.